And Kieran Krishnan, the CEO of Microbiome Labs, who joins us tonight. He's a microbiologist and a clinical researcher, has more than 15 years of experience in the dietary supplement and nutrition industry. Oh, we're gonna a little dinosaur there.
Right, yeah.
Darn velociraptors. Okay.
Right, yeah. They get out.
He spent several years doing hands-on research and development in the fields of molecular medicine and microbiology at the University of Iowa, and he has also designed and conducted more than 10 clinical trials as a partner in a contract research organization called LiveSmart Inc. And for the last decade, he has focused his knowledge and experience towards developing unique and effective nutritional products for other well-known brands in the dietary supplement market, focusing on why bacterial spores, not the bacteria found in conventional probiotics, make the most effective probiotic products. So please welcome Kieran Krishnan. Welcome, sir.
Thank you so much, and thank you so much for having me again. In discussing with Tamara about the program this evening, I wanted to kind of get your feel for what would be of highest value to the audience. I do a lot of work on leaky gut, and I wanted to talk about leaky gut and the role that spore-based probiotics play in leaky gut. But is there anything else that you wanted to make sure I touched on?
Dr. Carter, Dr. Harshfield, perhaps that is a question better posed to you two. What do you think?
Well, I mean, yes, and you do some very in-depth presentations, which are fabulous, right?
Right.
So, but I would just, I mean, our audience is very sophisticated. But again, I would say, really just kind of bringing up on the up to speed on your kind of your product line, because they all know what leaky gut is and all of that stuff. So, but your products are definitely unique in the market, even though there are other spore forming probiotics out there. So, and now that you've expanded to quite a robust line, I would say kind of giving them a flavor for what microbiome has become. Yeah.
Okay. Excellent. Well, that works. Thank you so much. So let me share a couple slides, and then I'll share some information through the slides if that's okay, and then we could chat as well beyond that. So there's one slide in particular I often walk myself through, and the audience through, and this becomes really kind of the basis for the vast majority of the products that we create, right? We call the intestinal epithelium the ground zero for most health disorders. And the reason why we do that is because, I've spent the last 10 to 12 years looking at literature. Of course, we've done a lot of our own clinical trials as well, really trying to dig into what are the origins of disease. And when you start to realize that the microbiome plays a significant role in the pathology or the origins of disease, then you have to ask yourself, how does the microbiome create a condition in which disease can occur? And that is, of course, in a dysfunctional microbiome, because a functional microbiome is the most protective and healthy thing you can possibly have. But a dysfunctional microbiome not only is not protective or healthy, it actually works against you in a very significant way, right? And that's the important thing for people to understand about the microbiome is it's a significant spectrum, where on one end, it's really beneficial, it's helpful, it's the reason we can exist today in many ways. And then on the other end, it's actually the most toxic thing in your system or that you're exposed to. So a lot of people are somewhere on that spectrum. Some people have gone to one or the other extremes. But at the end of the day, what this tells us is that we really need to pay attention to certain aspects of the microbiome in order to really understand health, wellness, and outcomes. So we always move to the intestinal epithelium. For those of you that may not be familiar with this structure, it's basically the mucosa layer with the intestinal epithelium, which is one cell thick, right? So this final barrier of things entering from the outside of the body to inside of the body, because keep in mind when things are in your digestive tract, it's still considered outside of the body because it's a tube that's open on both ends. And so unless something passes through your mucosa layer, past your intestinal lining cells, and get into circulation, it's not really inside your body yet. So the barrier for things entering inside your body are these two mucosal layers, and then also your intestinal epithelium, which is one cell thick, as I mentioned. Now, as you can see in the intestinal mucosa, there's two distinct structures. There is this mucin 2 layer on the inside here. This mucin 2 layer is basically a thicker jello-like structure that, as you can see here, is basically devoid of microbes. The upper part is more the mucin 1 layer, which is where most of the microbes exist. It's a little bit more fluid and more liquid-like layer that the microbes inhabit. And then down here, the intestinal epithelium is that one cell thick layer of cells sitting shoulder to shoulder. In between these cells are, of course, the tight junctions, and embedded within the intestinal epithelium are a number of functional cells, cells like M cells, L cells, Paneth cells. They make up a massive component of your immune system. So these are all immune cells, and then there are cells like goblet cells that produce things like that eat short chain fatty acids and produce things like the mucin layer, right? So the intestinal epithelium is a very defined structure this way. Now, this as it's illustrated, is a healthy intestinal epithelium, right? So and lining of the gut, because what you have are the two distinct mucin layers, most of the microbes up here in the top layer, and you've got this barrier system of mucin 2, tight junctions are all tight and sealed, and you have basolateral circulation down here. Now, this is all maintained by a high diversity of microorganisms in the mucin 1 layer, and then, of course, production of things like short-chain fatty acids and other postbiotics that rebuild this structure continuously, and then also having well-formed tight junctions. Now, most of all of this is controlled by the microbial system. So all the microbes within your gut microbiome play the biggest role in controlling the status and the structure of this intestinal epithelium and digestive tract lining system. Now, if your digestive tract is dysfunctional, it starts to look like this. And what you tend to have is microbes that eat away at this mucin 2 layer as an imbalance to microbes that rebuild the mucin layer. So what you tend to get is a net reduction in the mucin 2 layer, which means that this more liquid layer up top is going to fold into or translocate into where the mucin 2 layer is supposed to be. Now, if that happens, this creates a panic signal for the immune cells that are within the intestinal epithelium. That panic signal tells these cells that we have a massive influx of microbes coming through. This looks to them as if it's sepsis. And so the immune cells in the intestinal epithelium react accordingly. And so what they do is they start recruiting a whole bunch of immune cells and using inflammatory markers like IL-6, TNF-alpha, and so on to come to this region. So they start recruiting dendritic cells and macrophages and basophils and eosinophils and all these immune cells. They start coming to this region prepared to protect the host. And when they get there, because they're all innate immune cells, they start eliciting a massive inflammatory response. The problem with that inflammatory response is it's going to start damaging these intestinal epithelial cells as well, including the tight junctions in between them. Now, if that's happening and these cells are dying off and you're losing the tight junctions, you're going to start seeing gaps within these cells. And when you see gaps within these cells, you get a major influx of many compounds, including arguably the most egregious, a compound called LPS. LPS is lipopolysaccharide. It's made up here in that mucin 1 layer by about 50% of the commensals that live in your gut. And those that LPS is released continuously in this mucin 1 layer. It's released when microbes split and divide. It's also released when microbes die, and so microbes are continuously doing this anywhere where they live, especially in the GI tract. And when they release the LPS, you have free LPS floating around up here. Normally, in a healthy gut, what happens is it gets pushed up and moves out through defecation. But in an unhealthy gut, a gut that has this type of leakiness set up to it, you'll get a translocation of the LPS past the intestinal epithelium and now entering into circulation. With LPS in circulation, it becomes one of the biggest drivers of disease pathology. I do a whole 2-hour lecture showing all of the disease pathology connections between having LPS in circulation, elevated levels of LPS in circulation, and then also how that leads to the vast majority of chronic illnesses, right? So we're talking about everything under metabolic syndrome, things like autoimmune conditions, cancers, immune dysfunctions, and so on, including neurodegenerative conditions like Alzheimer's, Parkinson's, dementia, anxiety, depression, mood disorders. All of those things right now are driven in large part by endotoxemia, which is a term used for when LPS is allowed to leak through and end up in the blood, and then those conditions. So as a company, a lot of what we do are focused around helping the system rebuild this process and rebuild the mucin 2 layer, rebuild the mucin 1 layer, rebuild the intestinal epithelium, reestablish the tight junctions, and also then, most importantly, correct the balance of microbes that are up here in the mucin 1 layer. Because at the end of the day, the reason why this happens is because you have dysbiosis, which means you have an imbalance of microbes that eat away at the mucin 2 layer versus microbes that rebuild, right? And typically, that type of dysbiosis is measured as having low keystone species. So these are really important bacterial species that basically control the microbiome, they maintain diversity, they produce all kinds of compounds like short chain fatty acids that help metabolic processes, help reduce inflammation, help detox the body. They also produce other compounds, postbiotics, that help grow the other species that live within the gut so they can increase the diversity of the microbiome. And they do all this while maintaining a competitive structure so that outside organisms have a harder time invading and making you sick. So some of these organisms are Akkermansia, Fecalum bacteria, Prosnitzi, Bifidolongum, Bifidobacterium, and so on. And a loss of these keystone species will lead to a loss of diversity. And alpha diversity within the gut microbiome is one of the most important measures of overall gut health. We then start seeing a reduction of things like short chain fatty acids, and then we see this disrupted mucosa, disrupted mucosal immune response, and then finally the barrier gets disrupted and things leak through. So if we can work in this space and help all of these different components of the intestinal lining, then we're really doing a significant amount of service to people's health. And this plays a foundational role in the vast majority of conditions. So this is where we've really practiced. And at the end of the day, it's about reducing the amount of LPS and other toxins leaking through. So let me run quick then to what we call the total gut restoration. And I'll talk about the products within that, within this system, and then I'll mention a couple of the other key products that we work with. So when we look at that kind of dysfunctional gut, right, where the mucin 2 layer is eaten away, the mucin 1 layer is translocated in really close to the intestinal epithelium, all these microbes are flowing through, there's all this inflammation in the gut, the tight junctions are broken up, the intestinal epithelium cells are dying, so there's massive gaps, and you get leaking in of all kinds of things, especially LPS. When we start looking at that as key disease pathology, we have to understand that to really service the vast majority of conditions, you have to readdress that problem, right? So we do it in a couple of different ways. There's there's a 3-step system called total gut restoration, and that's what I'm going to talk about now. And then I'll mention a few other products that become important to think about when you're looking at overall rebuilding of the gut. So the first step in all of this is reconditioning the gut, right? Because we have to fix the microbes in order to fix the gut, because it's the dysfunctional microbes that are driving the dismantling of the gut lining. If those dysfunctional microbes still persist, you simply cannot fix the gut lining, right? So it starts with reconditioning what the gut microbiome looks like. That means increased keystone species, increased diversity, and competing against dysfunctional or opportunistic organisms. We do that with the Megaspore biotic, the first multi-spore probiotic on the marketplace. I can tell you when we came out with it in 2013, nobody knew what a spore probiotic was, right? It wasn't even a category in probiotics. And then now it's actually not only a category, it's actually the fastest growing category of spore probiotics. And I'll talk in a little bit about why we came up with it and what was the original idea behind the product. But here we use a Megaspore first in the first 30 days to start reconditioning what the microbiome looks like. And then Megaspore also on its own seals up the gut lining pretty well. And I'll talk about how it does that and show you the initial data on that. But once we start reconditioning the gut, we're starting to see a change in the gut microbiome. What we want to do is reinforce these positive changes, because throughout the day, based on all the things that you expose yourself to and you consume, we have exposure constantly to things that try to dismantle our gut microbiome, right? So whether it's gluten that you're exposed to, or Roundup in the environment, whether it's in the water, food that you're eating, all of these things have a negative impact on your gut microbiome. So we want to continue to reinforce positive changes that happen, and we use a precision prebiotic for that. In our case, it's called Mega Pre. These are prebiotic oligosaccharides. What we've been able to show ourselves, and then others have also published on these particular prebiotics, showing that they have a unique ability to specifically increase the growth of keystone species. And that's really what we want to get at the end of the day. If the keystone species are high, then they protect and create the rest of the microbiome and take care of it for us. So we want to start getting to that level where our keystone species are increasing in their numbers. So we use the prebiotic in the second month with the probiotic. So you stay on the probiotic in month 2. Now, not only are you reconditioning your gut, your microbes, you are now reinforcing those positive changes in a very significant way. And then finally, by the start of the 3rd month, you've got new microbes taking control of the lining of the gut. You need to provide them with tools for them to rebuild those important structures like the mucin 2 layer, like the intestinal epithelium, and so on. For that, we use a rebuilding tool we call Mucosa. In there, we have immunoglobulins, polyphenols, and amino acids. And to explain why we have those 3 major components, basically, these are the key building blocks of the mucosal layer, right? So polyphenols are used by an organism called Akkermansia muciniphila. Akkermansia muciniphila is a key organism in stimulating the rebuilding of the mucin layer. And if you feed Akkermansia the right type of polyphenols, you'll get a proliferation of Akkermansia. They will turn on a gene called the MUC-T gene, and they will also produce short-chain fatty acids, all of which goes towards rebuilding that mucin layer. We also use immunoglobulins, and the reason for that is if your mucosal layer that exists is toxic and has high degrees of inflammation, it becomes very hard to repair it, right? No different than if you got a cut on your arm or your hand. If you just left it, it would heal itself. But if you got a cut and every single day you rubbed it and irritated it, it would be red and irritated, and it would take far longer to heal. Same thing that's occurring in the gut, right? If your gut lining is inflammatory, it becomes so much harder for your gut lining to heal and get repaired. And that's why the immunoglobulins play an important role, because they have phenomenal studies on very complex gut inflammatory conditions like ulcerative colitis, especially ulcerative colitis in pediatrics, and then also HIV and HIV enteropathy. HIV enteropathy is a severe leaky gut type of condition where the NIH itself has published studies showing that HIV enteropathy and the degree of leakiness of the gut was a better predictor of HIV mortality than viral load itself, right? So this is a very significant driver of disease progression. And IgG, these particular immunoglobulins, have a couple of very well done published studies by a world-renowned HIV researcher showing that this product can actually dramatically reduce the inflammatory response going on in the mucosa, thereby allowing the mucosa to rebuild. In the ulcerative colitis study, they showed the same kind of thing, and this was in pediatric cases. What they showed was that because it can reduce the inflammatory response in the mucosal lining, they were actually able to see a regression of the grading of ulcerative colitis in the patients. So it was really quite profound when you have a product, an ingredient that can do that. And then the amino acids we're talking about, these are 4 key amino acids that are building blocks of the mucosal layer, right? So the mucosal layer is made up of amino acids and sugars, right? So these amino acids and sugars basically form a gel-like structure, and the 4 main amino acids that make up the mucosal lining are not necessarily present easily at adequate amounts in diet. And there are studies that show when you add in these 4 amino acids into a damaged gut, it increases the synthesis of mucin by over 95%, right? So this becomes the set of tools that your new microbiome needs to rebuild that whole structure. And this is what each of those products look like. Megaspore, was our first flagship product, launched back in 2013. We were the first to bring the spores to the marketplace, and certainly a multi-spore consortium product. Give you a brief history of where the product came from. As a microbiologist, prior to starting Microbiome Labs, I had a research company, and I was designing and running numerous clinical trials for other companies within the supplement space. And I also was consulting for a lot of companies within the supplement space, trying to bring some science to the products that they produce. There was a large multinational branded company that hired my research company to really do an analysis on probiotics in the marketplace, right? To help them figure out what is the direction they should be taking with probiotics. Should they be going in the refrigerator? How many billions? How many different strains? How can they have a competitive advantage? Through our research, one of the first things we did is we took 40 of the top selling probiotic products on the marketplace, and we put them through simulated gastric solution to see could any of them even survive. There's varying doses all over the place, but it doesn't matter if they don't get to the site of action alive. So we tested them, all of them failed, all of them would die through the gastric system, right? So in our mind, okay, there's just a lot of dead bacteria going through. Then when you start looking at it deeper, there are some strains, and they're very unique strains, where even the dead versions can have a stimulatory effect to the immune system, but the vast majority of these probiotic bacteria that are getting killed in the gastric system will simply just move through, right? It becomes very expensive poop at the end of the day. And so we saw that as being a big problem, and we wanted to resolve that. So we started researching what type of strains have a natural capability of making it through the gastric system and getting to the intestines alive. We wanted to see if there was such a species that had that capability. And sure enough, we came across Bacillus as a genus, and then a number of species like Subtilis, coagulans, and so on. And what we started to learn and realize immediately was that Bacillus is also a commensal organism in the gut, right? It exists naturally in the gut, and it also exists naturally in the environment. So this is one of those unique bacteria that has what we call a biphasic life cycle, meaning that they have 2 different phases to their cellular life cycle, one that's in the body, which is a very different environment than the one that's outside of the body, where there's UV light, there's desiccation and all that. And then inside the body, there's no UV, there's no oxygen in a large part of the GI tract. It's dark, but it's moist and all that, right? So very unique strains. We found Dr. Simon Cutting of Royal Holloway, who is a global leader in spore education and spore research. So we partnered with him, licensed these strains, and immediately started testing them to see what their efficacy would be. Today, we have 3,600 probiotic spore strains in our library, right? So 3,600 Bacillus endospore strains. We still only use 5. We are in development of a couple others, but what that really tells us is that most of these strains are not created equal, right? Just because it's a spore, it doesn't mean it's necessarily going to work. We are finding, as we screen our thousands of spores that we have, very few of them have probiotic-like functionality. It has to be a spore that evolved in the human GI tract without much time of evolution outside of the human GI tract, right? If not, it will adapt to changes in the environment outside the human GI tract. And so, it's important to keep that in mind because, as Dr. Carter mentioned, there's other spores coming out, but I would ask them to prove to you that they have efficacy by demonstrating or showing clinical trials on that finished product, right? So that's a very important thing because what we see when we test even other spores is many of them die, they don't necessarily survive, and so on. But that's Megaspore in a nutshell. Then we have the prebiotic that I mentioned, which increases keystone species in alpha diversity quite dramatically. And when you add Megaspore and Megapre, it actually has a tremendous synergistic effect where it more than triples the impact of the probiotic itself. That's called a synbiotic. That's how you define a synbiotic these days, actually, is that it's a prebiotic that enhances the functionality of the probiotic. When we look at things like short-chain fatty acids, we saw a 50% increase when you take just the probiotic, but we see almost 150% increase when you take the probiotic and the prebiotic. The Mega Mucosa, again, is the rebuilding product. It's our second best selling product in our entire line, which speaks a lot to its efficacy and people talking about it and practitioners talking about it. Mega Mucosa can make a significant difference in most people's gut if you have any inflammatory condition. Just to show you real quick, this was the first study that we did. This was a pilot study on Megaspore. What we did is we took, I think in this case, it was 30 or 40 college-age kids to look at their leakiness in the gut. And basically, if you find leakiness in the gut, what you see is that if you have them come into the lab at time 0, right, in a fasted state, and you measure their serum LPS levels, you'll see some small basal level like this, right? And then you give them a high-fat, high-caloric meal, and then what you should see within the first 5 hours is this massive increase in endotoxemia over that 5-hour period. And typically what we look for is almost a 6x increase in the level of endotoxin in these individuals, right? So we need to see this massive increase here. I don't know what happened to my little pen. Not sure what happened to the pointer. But what we tend to see is this 5 to 6-fold increase here between the baseline, which is the fasted state, and then the 5 hours after eating. And what that indicates is a massive influx of flood of endotoxins into your circulatory system, right? So these endotoxins are the ones that are generating a tremendous amount of inflammation within the body. So here. So this is what we ended up seeing after 30 days of putting them on the probiotic. This is the first study we published, where we saw at the baseline itself, we saw a reduced amount of total LPS, which means throughout that 30-day period, they were getting a lot less LPS leaking through. And then when we put these same patients under the same challenge where previously they saw this crazy increase in endotoxins, we now have this response where they're seeing almost completely flat endotoxin response, meaning nothing is leaking through into their circulation. So in these individuals, we saw almost a complete resolution of the leaky gut. And what's good about it is we saw also a complete resolution of things like a number of inflammatory markers as well. I'll show you just a summary of those inflammatory markers. So these in particular, these are, erase all this mess I made here. Okay. These in particular, these inflammatory markers indicate significant amount of distress in the body, things like IL-6, IL-8, MCP-1. These are all disease-associated inflammatory markers. If you look at conditions like metabolic syndrome, autoimmune disease, Alzheimer's, Parkinson's, all of these conditions, they have elevation of all of these markers. In fact, when you look at COVID itself, there was a study called the Boston-Dublin study. And in the Boston-Dublin study, what they were looking for were markers that could indicate, that could be predictive of people that will have a negative response to COVID. And out of all of the markers that they tested with individuals between thousands of cases in Boston and thousands of cases in Dublin, they found one that was very significant and always elevated and was predictive of a severe response to COVID, and that's this marker IL-6, right? If IL-6 is elevated, you have a much higher risk of having a really bad response to COVID compared to age-matched individuals that had reduced IL-6. Now, what's interesting about IL-6 is it's also the biggest driver in a whole host of chronic conditions. And one of the beautiful things here is if you know how to read a heat map, basically what we demonstrated here is that in these individuals, the IL-6, this is a spore treatment group, compared to these individuals here who were the placebo group, we saw a very measured reduction in IL-6 chronically, right? So a heat map, as the number increases, it tends to become more orange and red. And as the number decreases, it tends to become more yellow and green. So that's what this heat map indicates. So we were super excited about this. And this is just with Megaspore alone, showing that we can alleviate leaky gut, we can bring down all the inflammatory markers associated with leaky gut. And then we jumped into studies with the other two components as well, the Megapre and the Megamucosa. I won't bore you with all the studies, but we're more than happy to send them along so you can review them. So these are some of the studies when we combine them and we show a very significant increase in things like alpha diversity, keystone species, and so on. So let me go towards the end here. I want to share a couple things. So this is the Megamucosa. Here's some of the studies I talked about in HIV, in ulcerative colitis, showing the ability of the immunoglobulins in Megamucosa to restore function in the lining of the gut and reduce a significant amount of the enteropathy and the inflammatory response that are going on in this gut in very serious conditions. We also showed that the polyphenols not only increase short-chain fatty acids, but it can also significantly reduce inflammatory responses in the lining of the gut. And of course, these are the 4 main amino acids. This is a study that shows an increase in mucin synthesis by 96%. One of our other super exciting products is the gut-specific fish oil. We don't talk about it enough, and I'm not sure why. I think maybe because we were out of it for such a long time, but it's back in stock now. We have plenty of stock, and we were so excited about this particular product because the spores, the Megaspore, the Megamucosa, the Megapre, all of them take care of one massive chunk of inflammatory damage, and that is LPS-mediated inflammation or basal inflammation as well. This is inflammation that's governed through pathways like IL-6, IL-1 beta, TNF-alpha, and so on. As I mentioned a few times, all of those pathways and all of that type of inflammatory pathology is a major presence in chronic illness, right? So it's very important to address that. But then there's a second inflammatory pathway called the arachidonic acid pathway, right? So this is the prostaglandin pathway that leads to the expression of cyclooxygenase or lipooxygenase, which increases both inflammation and even pain. This whole system is mediated in many ways through what is happening in the gut through a condition called, or through a marker called AA-EPA ratio. And lots of studies show that high EPA fish oils tend to reduce mucosal inflammation. They promote goblet cells to differentiate, and then they modulate the intestinal microbiota. And then at the end of the day, they reduce that arachidonic acid type of inflammatory response, which has become really prevalent in conditions like breast cancer, insulin resistance, depression, acute coronary syndrome, colon cancer, and so on. This type of inflammatory marker called AA-EPA ratio has been shown to be extremely relevant in many of these inflammatory conditions. And basically what it's looking at is the ratio between arachidonic acid, which you see here in this pathway, is coming down from linoleic acid and alpha-linolenic acid. Arachidonic acid turns into a bunch of inflammatory and pain type of signaling markers, right? And if you have higher levels of EPA than you do arachidonic acid, then you negate all of these inflammatory responses. So what we were able to show is we can reduce AA-EPA ratio by over 40% in 8 weeks in individuals. And if you can do that with EPA, if you can reduce the ratio of AA to EPA, if you can increase the EPA, you can substantially bring down these markers as well. We also use DPA in this product. DPA increases the absorption of EPA and DHA, and we also use these very exciting compounds called PRMs, or pro-resolving or pre-resolving mediators. So basically what these are, if you look at fish oils and you look at things like the EPA and DHA of fish oils, in order for them to work at the cellular level, they have to be reduced all the way to these other fatty acids. And then these are the fatty acids that end up working in the cell to reduce inflammation. You know, this happens with fish oil, it's perfectly fine, but in some individuals, this process can take longer to happen. And in other individuals, depending on what SNPs you have, it may stop at some of these levels, right? So the fish oil may not be as functional for you. Now, when you put in PRMs, it means we're putting in this level, this layer of omega fatty acids that are already resolved and are only one step away from cellular function. And so when you add a defined amount of PRMs to a fish oil product, it means that the anti-inflammatory effect of the fish oil starts occurring much faster, right? So these to us were extremely important because when someone takes the product, we want them to start immediately getting that anti-inflammatory effect in the gut lining. And that's what you tend to see. So we were the first, and for a while, the only fish oil on the market that had PRMs, and then also that had higher levels of EPA than DHA. And then we also utilize a DPA in order to increase the absorption of both EPA and DHA. So I cannot overstate the importance of this particular product in terms of taking care of that whole other pathology of inflammation, that arachidonic acid pathway, which is so common and prevalent and a big driver in many, many different conditions. So this is an exciting product that we talk about all the time now. Now that it's back in stock, and we're excited to have it back in stock. Let me move on to the next slide. So for a huge majority of patients that come in, obviously the majority of people that are seeing functional medicine doctors have something wrong with their gut, right? And I think most people, most educated, sophisticated people are already attuned to that. And certainly the vast majority of functional medicine doctors are also attuned to that. We always recommend for most patients to start on the 90-day total gut restoration protocol. That's a great start because we have to start revamping all those different aspects of the gut mucosa, the lining of the gut, the microbiome, and then start rebuilding the structures in those individuals so that they can actually have stable structures in the lining of the gut. A couple other products to consider while you're doing that. If it's an individual that has lots of mold toxicity, for example, or is highly sensitive, or tends to have a really profound inflammatory response in the gut, meaning they're dealing with things like Crohn's or colitis or microcolitis, they're dealing with these kind of inflammatory gut conditions as part of their total picture, it would be great to start these individuals with the IgG when you start them on the Megaspore as well, right? And with the Mega IgG, I always recommend doing 4 caps in the morning when you wake up as on an empty stomach, and then 4 caps in the evening after dinner. This product works phenomenally well. So for that subset of individuals, it becomes important to add the Mega IgG early on. And then, of course, any sort of inflammatory response that people tend to have, the fish oil plays an amazingly important role in that. We had great success throughout COVID with the fish oil for people that were going through, hell and high water with not only the disease pathology, but being stuck at home and, experiencing all kinds of dysbiotic drivers at home, like over-sterilizing and medications and all of that. This omega fish oil plays a very significant role. Other things to consider for patients that are dealing with fungal overgrowth, so like Candida overgrowth, we have a product called Mycobalance, and we developed this product because we saw fungus and mold in the gut, especially in the gut microbiome, and then other areas distal to the gut, as a major indicator of dysbiosis that's occurring, because these are opportunistic organisms, right? They proliferate and they grow where microbes are weak or microbes have been eliminated, whether it's from antibiotics or other reasons. So when you see overgrowths of things like Candida and fungus and of course mold in individuals, what you're really seeing is a severely dysbiotic microbiome that's allowing for the opportunistic growth of these organisms. So in order to repair that system, what you really need to do is not only kill off the surface microbes, the surface fungus and mold in particular, but you also have to go in and interfere with the barbing root that these organisms have. So, if you're familiar with how a mold grows on tissue, not only is there surface growth, but there's a barb that sticks into the tissue that basically anchors it down there. So you could end up killing the surface microbe, but the barb is still there acting as a root. So we formulated a very unique product with undecylenic acid and B-propolis that actually takes care of both of those in these individuals. And if the individual has the issues we talked about before and a fungal overgrowth, they can start this product at the same time as the other one. Another very, very innovative product that we're super excited about is Piloguard. Piloguard is a ghost probiotic, meaning it doesn't have to be alive. It's actually an attenuated probiotic strain. It's a completely natural strain that was derived from human volunteers, so we know it's a strain that works and functions in the GI tract. What it does, and it does only one thing, which is what's so fascinating about this strain, is it grabs onto H. pylori in the stomach and takes it out of the system. Right, that's the only thing it can do. And keep in mind, it's not alive, right? So its whole cell structure is there, but it's not a living cell. So even the dead version of this bacteria has a affinity for H. pylori on its cell membrane, which means that when it gets close or bumps up into H. pylori, it aggregates to it. And then what you start to see in electron micrographs, which I don't have here, but we can visualize all of this, you can see these bacteria bumping up against and grabbing onto Helicobacter pylori, where they form this co-aggregate where you have one H. pylori and 4 or 5 of these microbes surrounding it, and it takes the H. pylori out of the system through defecation, right? It doesn't harm or touch anything else. This is the only thing it does. And I think we have 9 published human studies on this, I think with close to 1,000 patients on this product. And basically why you would need to consider this, right? So H. pylori is probably the most common pathogen. The lowest estimates are that at least 50–51% of adults have infectious levels of H. pylori. Most people who have it are infected early on in life, like around the age of 7 or 8. And it typically happens in households through exchanges of bodily fluid like saliva and so on, if you're sharing cups and utensils and all that within the household. From the beginning, once H. pylori settles into your system, it's likely one of the most profound drivers of dysbiosis. And here's why I say that, because what does H. pylori do? It gets into the stomach, it bores itself into the stomach lining, it sits right at the gastric epithelium, and it produces all kinds of toxins that eat away the gastric epithelium and also drives a significant amount of inflammatory response in the lining of the gastric system, right? And that inflammatory response has a few different outcomes. Number one, that inflammatory response can burst into circulation and drive inflammation throughout the body. This is why Helicobacter pylori infection is associated with systemic cancers as well, right? It's also associated with autoimmune disease. And it is thought to be a driver of both of those conditions because of this bursting through of systemic inflammation. The other thing it does is it creates significant inflammation in the stomach itself, and then it uses a buffering solution, urea in particular, or urease, in order to reduce the acidity of the stomach, so increase the pH of the stomach, because Helicobacter pylori prefers the stomach to be less acidic than it naturally is. Now, once it creates...
Karen, Tom Lewis here. Can I ask a question about that whole thing?
Yeah, absolutely.
First of all, how does the gut make acid? Is it a just-in-time type situation, and what organs really play a role in that?
Yeah, so acid is actually made in the stomach, and acid is made in response to a couple of different stimuli. As it turns out, histamine is one of the key stimuli for acid production. And then the other stimuli for acid production are micronutrients like zinc and magnesium. These are the important stimuli to create acid in the stomach itself through glands, through your glands within the stomach that are the HCl producing glands. The other things that trigger acid production is the action of chewing. So one of the things I've always recommended to people is to not chew gum all the time, because the action of chewing actually causes your stomach to start to produce acid. And when it's producing acid, when you have no food in there, you're actually burdening the glands that are the acid-producing glands. And over time, you can burn them out. You know, just like your pancreas burns out and you end up with pancreatic insufficiencies later on in life where you're not producing enough digestive enzymes. You know, the chronic use of antihistamines may have a role in reducing stomach acid production as well, because histamine being one of the key signals for acid production in the stomach. And
interesting.
Yeah, and Helicobacter pylori
is there a number 2 organism to Helicobacter pylori in
as
your
as a pathogen?
Yes.
Number 2. So I think Helicobacter is probably the biggest driver of dysbiosis and inflammation. I would think that it could be a tie between a couple of different organisms, one being Bilophila wadsworthia, and we're starting to see more and more of that opportunistic organism in the Biome Effects test. And Bilophila wadsworthia is a bile-loving organism that lives in the small intestine, but it typically indicates a significant amount of dysbiosis in the small intestine, and it's associated with things like SIBO and other inflammatory conditions in the small intestine.
Is it an archaebacteria that's producing methane?
Yes, it can produce methane, exactly. Yeah. So it has that capability. It's doing some fermentation in the gut, in the small bowel, which is not good, obviously, for the small bowel. And we feel that as a response in bloating and things like that. It can also, I believe it's a sulfate-reducing bacteria, which means it can take sulfates from foods that are coming into your system and convert it to hydrogen sulfide, which increases inflammatory damage to the large bowel. So from the small bowel, it can create a bunch of inflammatory damage to the large bowel as well. And then Clostridia is probably a bit of a tie for that, because people are so often exposed to antibiotics and all that. Clostridium difficile is a key opportunistic microbe. Once it establishes itself and it starts producing its toxins, it becomes a difficult thing to control, and it leads to long-term inflammatory damage to the large bowel.
And you have a product. You have a product for that. Let me make one more comment about the H. pylori. I think Quest made a mistake of no longer doing blood labs, because it lives in the gut lining, so it's in the blood. Yep. And LabCorp thankfully doesn't say H. pylori less than, if it's below their reference range. They actually give you an absolute number, but the number that they give for a reference range is really titrated to stomach ulcers.
Yeah. But
your point about all the other insidious things that H. pylori does, particularly release the urease that converts nitrogen compounds to ammonia, which, you know, buffers the acid.
HCl, yeah.
So what I found is, you know, I kind of use the term, you know, you can call it gut dysbiosis, but I also call it gut resistance, kind of like insulin resistance. So in this context, when H. pylori is even at 1/3 or 1/4 their reference range, if you have gut resistance or gut dysbiosis, it can still play a major role in impacting overall gut integrity, because, you know, the acid and the organisms live in a symbiotic environment. You have to have both.
Absolutely.
And so that's so like, I think it's 0.79. I forget the units of measure. The LabCorp considers it negative in terms of pathogenic. But what I found is in very disrupted guts, as low as 0.2, still need treatment. You still need you want to be below that. It's kind of like your insulin is super high, and a little bit of sugar, you know, sends you into a tailspin.
Right.
Same thing with the gut.
Absolutely.
And the other comment I'll make for you is that over time, just running these tests over and over and over again, you know, there's no good real test for the gut.
Yep.
But a combination of sed rate, homocysteine, and ferritin iron tell me a lot about what's going on in terms of absorption.
Right.
So then you can titrate back and make an assumption about dysbiosis, because, you know, almost every functional doc puts people with elevated homocysteine on, you know, methylfolate and methyl B12. And what I've done is none of that, and just worked on diversifying the microbiome. And lo and behold, homocysteine always comes down.
Absolutely. Makes sense. And that, you know, one of the reasons I think for that is when you look at MTHFR gene mutations, right, the prevalence of it is so high in the population. Normally, genes that are critical and that have SNPs get worked out of the population, right? People that have those SNPs don't survive. The reason why it's less important is because the microbiome produces a whole vast array of methylated vitamins. So you're absolutely right in your thinking and approach that if I diversify the microbiome, it overcomes that shortfall in our own pathways. And a couple of the mentions on Helicobacter pylori, you know, it drives SIBO. There's a H. pylori induced SIBO that's present, right? We also now know that it plays a role in infertility. So all of these areas distal from the stomach and distal from the gut, it can impact both male and female fertility. And as it buffers the HCl and reduces the acidity in the acidicness of the stomach, one of the egregious effects of that is allowing more and more of the gram-negative bacteria in the oral cavity to survive through the stomach and enter into the small intestines viably, right?
I think that's how you get SIBO.
Absolutely.
You pass right through the gut because, I mean, the stomach, because the acid's insufficient.
Absolutely. And it's 100% because if you look at what is happening in the small bowel with SIBO, normally in a healthy small bowel, you should have a predominance of gram-positive bacteria. What you tend to see in people with SIBO is what we call a taxa shift to gram-negative bacteria, microbes like Enterococcus fecalis and Faecae. These organisms exist in high prevalence in the mouth, and they move down, and they're allowed to survive through. And that's why PPI use induces SIBO. There is a definitive condition called PPI-induced SIBO for that reason. Right? Yeah. And so, in our view, this and the thing is, when you have patients with H. pylori, even very low levels, as the test indicates, it becomes a difficult thing to think about trying to treat them with the triple therapy, right? Which is so harsh on the body, the broad spectrum antibiotics and the PPI. And so that's what we wanted to innovate, a microbial solution to it. And the 9+ studies that we have published on this shows that this Piloguard product brings down H. pylori just as well as any of the medications.
It makes total sense. And the triple therapy is, if you're beyond all hope and you know your gut's in such dysbiosis, that just get rid of the H. pylori. But it really is a last resort, and I almost never recommend people go down that path. I'm going to ask you one more question, then I'll shut up. The talk has been phenomenal.
Thank you.
Because this is the most important area in healthcare. But Dave Osprey bragged about, I have a video on one of my blogs about, he has 160 unique organisms and he thinks that's fantastic. What's your general opinion on the number of organisms that's optimal, and does it really matter because you have these most important ones that have to be there regardless of some of these less significant ones?
Yeah, that's a phenomenal question. So if we look at the last 10 years of microbiome research, what keeps coming up, the theme over and over again, is that alpha diversity is critically important. The keystone species are equally important. They should be there. They should be present. But if you have high keystones and low alpha diversity, you will still run into problems. You'll still run into metabolic problems, lack of certain functionality, right? Because that's the biggest part of our genome, is our microbiome. And unfortunately, you know, as Dave is a great biohacking guy and does all kinds of crazy things, but 160 species is just not great. We actually work with a number of researchers like Jens Volter that have spent the last 6-7 years studying the Papua New Guinea tribes, and other microbiologists that work and spent time with the Hadza tribe. These individuals indicate to us where our microbiome was, right? When we were hunter-gatherers not that long ago. They have over 350 viable species within their microbiome.
Yeah, you got to go out to the backyard and eat more dirt.
Yep, exactly. And hopefully a
fecal transplants have up to 1,000.
Absolutely, yes. And you could have upwards of 13,000 different species in your gut microbiome, right? So, and what we're seeing, because we run the Biome Effects test, which is a whole genome sequencing test for the microbiome, what we're seeing over the last few years, and I think COVID probably, and all the things we've done for COVID, have probably played a significant role in continuing to bring down our diversity. Because the tests that we're running this year and the end of last year, we're seeing a trend of lowering alpha diversity compared to 2018, 2019. So, you know, it's a scary thought for me. That's the kind of thing that keeps me up at night about us losing this amazing reservoir of organisms that we're supposed to pass down to the next generation. And we're not doing it, you know, and if we don't do it, they're going to be faced with problems. One last product I'll mention is our Zen Biome product. This is a psychobiotic, 1714 strain. It's called Bifidolongum 1714. It's a strain that has an exopolysaccharide. What's so unique about it is not only has it been shown in 7+ published studies to dramatically reduce the perception of stress, improve mood, reduce the cortisol peaks from a stress response. It also changes brainwave function. So it puts people more in that theta wave state, which is that meditative state where you can rationalize things well, you're more calm, you can actually deal with stressors that come along. Now, what is so important about this is it does so by reducing the inflammatory response that cortisol and the flight or fight response in the body creates. And so it brings people out of that flight or fight response. And in fact, the flight or fight response drives neuroinflammation, because part of the flight or fight is getting inflammation and perfusion to the brain and the central nervous system to get the body ready to fight or flee. That's the same thing that's happening, for example, in COVID long haulers. These are individuals with neuroinflammation, and they have the same exact elevated markers as people that have chronic anxiety and depression. It's the same biomarkers, same inflammatory processes, and that's why a lot of long haulers tend to have neurological impact as a primary outcome. So we're actually doing a study now on the 1714 strain, looking at COVID long haulers and if we can help and support them in some way. But Zen Biome becomes a very critical part of the gut-brain axis. And what we tend to find is at least 52% of people that have something wrong with the gut will also have a dysfunction in the gut-brain axis, which becomes important to treat that component, you know, to take a complete holistic approach in helping these individuals. So yeah, I'll stop there. These are some of the key products I wanted to mention. Happy to take any questions. I'm sure we're also kind of running loose on time here.
Yeah, let's do that for questions. Guys, if you would, go to the reactions down below if you are on a desktop, and if you're on an iPad, I believe it's up above, but go to the reactions and choose raise hand. That way you will be able to raise your hand. And Andrew, you are first. Fire away, sir.
Yeah, no, that was brilliant, Kieran. And it's amazing. Every time I watch you speak, it's like something new or something different. You're always learning from you. It's great stuff. So thank you for your work and
thank you so
all
much.
of what you do. Yeah, I kind of was, I think you had started really getting into the H. pylori and low stomach acid medication, the proton pump inhibitors. But I'm wondering, is that the Piloguard that you would recommend if I have someone that had, you know, been on Prevacid or whatever stomach acid blocking medication for a number of years? Is that kind of where you were leading, or is that what I gathered from that? Would that be accurate to say? Is to probably to do all these things, but especially the Piloguard? Especially
the Piloguard. If you were on PPIs, absolutely, because with that reduced stomach acid, you're going to have H. pylori inhabiting your stomach and likely overgrowing as well. The vast majority of adults seem to be infected. And if you have symptoms like gastritis or gastroparesis, reflux, all of these very, very common GI-related symptoms, still some of the most common reasons why people go to see a doctor. If you have any of those, the beauty of the Piloguard is because it does nothing else but remove H. pylori. There's no safety issue in just treating it as if you had elevated H. pylori, right? Without having to go in and do urea breath tests and so on. And here's an important note: because H. pylori is transmitted in between people, within households especially, if somebody within a household likely has elevated H. pylori and all the associated symptoms, it becomes important for everybody else in the household to go through 3 weeks or 4 weeks of treatment to bring down the total H. pylori in that household, so you don't keep reinfecting each other. And the way Piloguard works is it's actually a capsule, so it's 1 capsule per day, but you actually pull open the capsule and you mix it in a glass of water and you drink it. And the reason we have it that way is with H. pylori, it's a bit of a contact sport, right? So we want to spread the Pylopath strain, the Lactobacillus plantarum, across the stomach lining so that it has a better chance of running into the H. pylori and grabbing onto it. So that's how we use it.
The other thing, Karen, is the H. pylori can start in the mouth.
Exactly. Yes, it
so
can.
People should probably swish the Piloguard. And Andrew, in my humble opinion, if someone's been on PPIs, whether they have or do not test positive for H. pylori, because most of the tests are inadequate unless you're interpreting it correctly like we do, you should throw the kitchen sink at rehabilitating the gut. It's not just about treating the H. pylori.
Yeah, well said. And then I guess someone else mentioned this too, is just like trying to maybe get the difference between Biome, which is, right, of course you're familiar with that, and then the Biome FX, which is the test that you guys have come up with. So
yeah, absolutely. I could do a whole hour dissertation on the differences.
I mean, I didn't mean to, yeah.
Let me give you just a quick snapshot, right? So I would encourage everyone to Google Biome and look up what microbiome and genetic researchers are saying about it. There's actually a whole blog by a very, very prominent, well-published genetic researcher that basically talks about the big problems around that test. It's a Silicon Valley funded operation. It's very much a black box technology, meaning that they don't ever disclose how they do any of the things that they do. And the biggest problem is that it's an RNA test, right? And the problem with RNA testing is that, number one, RNA is incredibly unstable. RNA is single-stranded. It's not designed biologically to be a long-term messenger. It's a single strand that goes from transcription to translation to make protein. And so it's a single snapshot looking at what RNA has been expressed and then captured in that particular stool sample. If you're not expressing certain genes, or you're not making certain proteins at that time, you don't capture that RNA. And if you don't capture it, does that mean it's not there? It doesn't, right? And then just the instability of RNA is the thing that really drives researchers crazy, because the interpretations that they make to provide the recommendations and all that, the science to interpret that from RNA does not exist. And so that it's highly problematic. You should Google and look it up. I think you'll find a lot of fascinating writings on it. BiomeFX is a DNA-based test, right? So DNA is double helix. It can be very, very stable. As we know, in crime dramas, they can pull DNA from things from 10 years ago and analyze it. And then also the biggest difference is that between BiomeFX and other stool tests as well is that we do the whole genome sequencing. Whole genome sequencing is basically mapping the presence of a microbe by sequencing its entire DNA from end to end. There's another technology called 16S, which is what most of the places use. The problem with 16S is it's looking for certain chunks of the bacterial DNA. It's only looking for 5 or 6 regions of an entire genome. And if it finds, say, 3 of the 5 regions, the algorithm will make a prediction of what bacteria that is. But studies have shown that that kind of methodology at the species level can be upwards of 70–75% error prone. So the accuracy is really poor. Whole genome sequencing is the way to go.
All right, before we get to Doris's question, Dr. Tom, Dr. Carter, if you could verify that most, if not all of these products are available through our full script, is that correct?
Yeah, so I'm full script. I've
Okay.
been prescribing these for many, many years. And yeah, Christian, yeah, you came up with a couple that I haven't been, privy to. So
Awesome.
those are really cool. Yeah.
Yeah. Well,
I want to point something out to Andrew, and the whole group, but I'm a medical scientist, and so testing is paramount. But Dr. Trent, my mentor, said, you test to treat. And in my humble opinion, we need to do, everybody needs to do a gut rehab prophylactically every year, independent of testing. It's just that important to know that we live in an environment that's going to be harmful to our gut. So just get right to it, roll up your sleeves, and work on your gut every year. Yeah. Even if you have ghost poops and the whole thing, everything seems perfect. I can guarantee you're not optimal. So that's kind of my stand on the testing. Sure, if you're really a mess, like I have a daughter that's a mess, and she's finally come to me, and we can test. But the thing is, treat with this program first, and then see if there's a resolution. Then, if not, maybe you do some testing. But treating in this particular case Karen, would you agree, would never be a harmful thing to do?
You're absolutely correct. And keep in mind, and I think, what you're saying actually speaks to this, that we are surrounded constantly by things that harm our microbiome, right? We didn't know that this microbiome existed even 10 years ago. So everything we've designed around us, all the chemicals and things that we have in our products that we use, our personal care products, our agricultural products, and our water, and the paint, everything, all of those things harm the microbiome. And one of the things I always say is that we are made up of microbes, right? We're this beautiful microbial construct. We're essentially a walking, talking rainforest, and we've taken this microbial construct and put it in an antimicrobial world. So we've designed around us an anti-human world, which means that we have to very diligently and constantly be fighting to maintain that balance, right? So I think you're absolutely right. No one can ever go wrong with trying to improve their microbiome, and I think it'll pay off in spades.
To that point, what's the impact of drinking municipal water with whatever it is, 0.3 ppm chlorine?
Yeah,
on the gut. Have you done any studies on that?
Yeah. So in fact, and it's exactly why I have a whole house filter with the RO system and all that to remove the chlorine and even the fluoride, right? So they started adding fluoride to the water because it was supposed to be good for the teeth, but fluoride in that format is actually an antimicrobial. Chlorine, absolutely at that level, is an antimicrobial. And the problem with antimicrobials like that is that they're low grade and they're persistent, right? And low grade persistent antimicrobials create very significant dysbiosis over time. And the reason for that is it's a continuous negating force on the microbiome, right? So certain microbes, what they'll end up doing is adapting to it, and then they can proliferate as long as that force is still there. And other microbes that don't adapt well to it will start to diminish. This is the same thing we've seen with glyphosate, for example, Roundup, right? Roundup and the active ingredient glyphosate is a antimicrobial. It's a powerful antibiotic. The problem is the shikimate pathway that it works on, a number of pathogenic organisms have a workaround for it, whereas a lot of your commensal organisms don't. So as you get exposed to low levels of that every single day, what you're doing is creating an ecological force that selects for pathogens. So you end up with this completely disrupted ecosystem where pathogens are prevailing. I'll just mention one study that we did that just recently published on a pediatric microbiome. We were interested in figuring out what is the impact of, say, 3 weeks of exposure of glyphosate to a pristine 3-year-old microbiome. This was an individual that was born vaginally, wasn't vaccinated yet at all, never had a course of antibiotics, was breastfed for a year, had a very healthy gut microbiome. And we exposed this gut microbiome to Cheerios level of glyphosate, right? So the level that is approved by the EPA and the FDA in cereal, in kids' cereal. And in 3 weeks, we saw changes to the gut microbiome that were equivalent to the types of changes you see in inflammatory bowel disease, right? So profound in that 3-week period. Fortunately, we added the spores in for the second 3 weeks as we were still increasing the glyphosate exposure, and we started to see a reverse of some of that because some of these microbes, like Bacillus, have found a way to metabolize glyphosate. So, we have to be active, and we have to be actively involved in maintaining our gut. It's a battle, it's an ecological battle that we constantly have to fight.
Kieran, Dr. Tom, Dr. Carter, we want to be respectful of Kieran's time. What do you say we maybe 5 more minutes of questions? Would that be all right? I know we have a couple people who have been waiting here. Doris, go ahead. You are next.
Already answered, but the one question I do have is, can we get the gut microbiome labs through Health Revival Partners?
Yeah,
what are the services?
It's all on Fullscript. All the products are on Fullscript.
Oh, are you talking about the BiomeFX test?
Oh yeah, so
get the test.
You can contact me if you want to get that microbiome test. Yes.
Okay, and someone already asked the question in the chat, but if you're doing this protocol, do we continue with the digestive enzymes and all of that?
I'll let the doctors answer that, but my opinion is absolutely, yeah. Digestive enzymes are always great for breaking down your food and helping your system assimilate the nutrients. I don't see an issue with utilizing digestive enzymes while you're trying to fix your gut.
Yeah.
Thank you.
Absolutely.
Yeah.
Dr. Carter, would it be all right with everyone if they wanted these products and they already know they need them to get them through Fullscript? Should they just contact Jody?
Yeah, contact Jody, or again, if you want more directed protocols and so forth, just contact me and we can go down that pathway. But yeah, these have been amazing. I use the products for myself and family and lots of patients, and just gotten rave reviews. And then just a handful of people who are quite sensitive that may need to start off slowly.
Yep.
Because it can, in some exert a Herxheimer reaction, but it's pretty low. Karen, one thing, I do have a couple of patients that noted that the prebiotic in the ingredients that said contains milk.
Yes.
So they were concerned about that, even though I know I'm sensitive to milk products, but I haven't had an issue with it. But that was something that came up, of course, on, like, let's say their food sensitivity test.
Yeah. Yeah. So unfortunately, that is one of the FDA rules, that if anything was derived from any component of dairy, even though there's no actual dairy left in it, you have to label it. So what it is it comes, the oligosaccharide does come from milk, but oligosaccharides are complex carbohydrates, right? So they are filtered and purified out. There's no dairy protein. There's no casein, whey, any of the dairy sugars. It's purified carbohydrate. However, and we've never had anyone with the dairy sensitivity have a response. However, in response to people asking lots of questions, we're actually I think next week maybe launching a version that doesn't have the one oligosaccharide in it that is derived from dairy, just for people's comfort, and so on. So we will have a variety that is acceptable for those people.
Good, excellent.
Yeah.
All right, Kay, you are next, and after Kay, we'll let Dr. Harshfield have the last word.
Thank you. Karen, for those of us with IBS who have had trouble digesting oligosaccharides and have had trouble with the Mega Prebiotic, can you please recommend something additional to feed those keystone strains?
Yeah, absolutely. So, and when you say trouble, Kay, can you describe a little bit what you experience as trouble?
I have food sensitivities, and I just couldn't digest it. I ended up with loose bowels and things like this.
Sure. Okay. And keep in mind that your system is not supposed to digest the oligosaccharides. Oligosaccharides are non-digestible carbohydrates. They're supposed to go to the keystone species in the gut that then transform them to things like short chain fatty acids. But a couple of strategies around that, right? So one is you can do very small doses and mix it in a lot of water and kind of sip it throughout the day, just to try to get some of it into your system. But looking at foods that contain high levels of oligosaccharides, there's a number of options as well. So polyphenols, even though they're not oligosaccharides, polyphenols feed Akkermansia, which is one of the keystone species. So if you look at polyphenols from kind of red or purple berries, even grapes, and somewhere around 500 grams or so of the food itself for polyphenols. So you can do blackberries, raspberries, blueberries, grapes, and so on. If you can manage those, if you could do 300 to 500 grams a day of those, it gives you enough polyphenols to help the growth of Akkermansia. Other foods, you can do resistant starches if you can handle them. So things like sweet potato, cassava, these things tend to have lots of oligosaccharides in them, and that's part of what will feed these keystone species like Faecalibacter prosnitzi or like Bifidolongum. You can also do certain things like beta glucans. Beta glucans can actually feed some of these, and that you get out of things like mushrooms, for example. You can also use those to feed some of these keystone species. And another counterintuitive thing, and this you have to talk to your doctor about if you can do, is doing some intermittent fasting. So, some 14, 15-hour fasting periods, most of it throughout the evening. If you can do that, that actually increases the growth of keystone species, providing your system with no food for a period of time because of the way the fermentation works in the gut microbiome. So hopefully some of those things you can add into your diet to start supporting the keystone organisms.
Yes, thank you so very much. I've been doing intermittent fasting. I know I'm thankful, and I'm going to try some of these others. Thank you so much.
My pleasure.
And one thing I wanted to interject before Dr. Harshfield comes in. So when it comes to testing, of course, I do a lot of deeper dive testing. And when Kieran was talking about, let's say, the lipopolysaccharides. So, I do the intestinal permeability test, which actually gives you an idea of your level of lipopolysaccharides, along with histamine, diamino oxidase, all of these things. So that's a really illustrative test when it comes to difficult GI issues. And I really, really like what I'm seeing with these products and helping to lower LPS levels and so forth. So great test to also get when when there are some difficulties with this, shall we say.
All right. Thank you, Dr. Carter. Dr. Harshfield, the floor is yours.
Thank you. That was awesome presentation. Thank you so much.
Thank you.
This is not so much landing the airplane as a submarine. We kind of think about our gut. It's like the ocean. We live on the surface of our body. We don't think about what lives in there. I mean, there's bacteria, there's fungi, this huge virome. I mean, and thank you so much for that organized approach to this huge, almost bottomless pit. One comment I would have, a lot of this stuff I think we discover is by accident. Like we talked about those histamine 2 receptors. We were monkeying around with antihistamines for the lungs, and somebody went, hey, wait a second, this stops hydrochloric acid production in the stomach. Oh, shoot, there's histamine 2 receptors. Okay, so here we go. So we're going to learn a lot. This is a perfect group to teach this to, by the way, because this is so safe. One little comment that I would have is that the SIBO, small intestinal bowel overgrowth, we had looked at, and this is a big hammer, by the way. I mean, what we're trying to do is not make this such a big deal, but the fecal transplants that we used to do, you alluded to that, and people with C. diff, we'd get someone in the ICU, we'd put them on a little antibiotic. Oh my gosh, C. diff is going to kill them. We'd do the fecal transplant with an enema tube. They started noticing that some of that transplant, it was great, actually reflux. There was an ileocolic reflux, like you'd have reflux of stomach to esophagus, gastroesophageal reflux. The valve down there, the ileum and the colon, the ileocolic valve, was allowing reflux to contribute. So we've got things going 2 different directions. I mean, it's really interesting. What is your experience? I know this is way bigger hammer than what we're talking about tonight. The microbiome transplantation doesn't have to be done with an enema tube. They have now got gel capsules. You take 30 of these, repopulate your gut. The thing about it, it's just like some of the little stem cell injections we do. We're doing something that's changing the epigenetics. We're not changing your DNA, but that can be generational. Those changes can change you and your family for 20 years. Kind of what is your take on the next level, the microbiome transplant and so forth?
Yeah, great question. So, early on, one of the big risks of the microbiome transplantation was you didn't know what you were putting in an individual, right? You knew that, like, for example, with C. diff, you knew that what you were doing was you were taking microbes from stool from an individual that did not have C. diff overgrowth, and then putting it in an individual that did have C. diff overgrowth to hopefully outcompete the Clostridium and alleviate the C. diff, but you didn't know what else you were transferring to this individual, right? That's the part that becomes a little bit of a iceberg. But now with the metagenomic testing that we have and the ability to go deeper into that type of the species level identification, you have a better line of sight as to what you may be transferring. There were some very famous cases on fecal transplants that basically altered people's body compositions and the way they walked and crazy things like that. That illustrated how important donor selection and the understanding of what all is coming with that fecal transplant needs to be, right? That's the focus. Now, the next generation, and in fact, I'm working on this with a couple of groups of people, where you can find because of the metagenomic testing that you can do now with such clarity, you can look at individuals with particular conditions and understand the functional groups of microbes that they tend to be missing or tend to have too low levels of, right? So certain types of phylums of microbes or even genuses or orders of microbes that they may have low levels of. And you can then actually take healthy donors and select out of the samples those particular phyla or that particular order or that particular genus, concentrate it, and then only transplant that component. So it's far more targeted and personalized transplantation, and that has a very interesting future because you are you're transferring a smaller amount of genetic material. You're transferring a smaller number of microbes. So you're reducing your overall risk of transplanting something that may not be good for the recipient. And you're doing it in a targeted manner that could have a very positive and quick impact for their microbiome. What we're also seeing is a temporary transfer of postbiotics that are made by those classes of microbes may be enough to increase the growth of those particular microbes in the recipient, right? Even without transferring the microbes, just what those microbes make in the donor. So that's another very exciting area as well. It's a fingerprinting of postbiotics and then utilizing that fingerprint and transplanting that into the recipients. So we're doing some of that with a couple of groups and companies, but there are probably much smarter people doing an even better job with it. But that is an exciting area to look into.
I think a lot of that started several years.
David, I'm not being respectful of time, but I'm just curious. I was having a thought about a transplant from a non-human that can digest things that humans can't digest. And would that be something in the future, or is that off the table?
No, it actually it's not. You know why? Because it actually happens in nature. So the zoonotic transfer of genetic capabilities for digestion especially has been demonstrated. I'll give you one example of that. There is an enzyme that's required to break down seaweed. The enzyme is called beta porphyrinase. Humans don't produce we don't have a gene to produce beta porphyrinase. And so we actually can't break down seaweed effectively. But what the studies have shown is that the Japanese population, their microbiome, and then maybe some of them have even developed the genetic capability of producing beta porphyrinase, but they gained that capability from eating raw, uncooked fish. So the fish have that enzyme.
I'm weird enough, Karen, to have read that a couple years ago.
I figured you would, yes. That's fascinating, right?
I want to be able to drink out of the same murky water my dog does.
Yeah. Yep, exactly. So there is that type of zoonotic transfer, and then there's also evidence. Of course, there's things called microRNA that can transfer from the plant kingdom to the animal kingdom, right? That affects our epigenetic expression. So these are, in some ways, some of the benefits of eating certain foods is not just the macronutrient composition of the food, but what microRNA may be in the food and how it alters our epigenetic response, right? So it's super fascinating. There's so much we don't know yet, and we're right at the tip of it. So it's an exciting time to be around.
Kieran, there are many questions that were asked, but of course we did not have time to answer. Is there an appropriate email address or some other method by which people could ask you these questions and get their answers?
Yeah, absolutely. I don't know if there's a way to copy and paste these questions from the chat. I think they're all in the chat. If it is possible to just copy paste, if you copy all those questions and send it to me, my email is these crazy initials that you see here, KRKH at Novozymes, so NOVOZYMES.com. If you send the questions that weren't answered to that email, I'll make sure to write down an answer and send it. And I'm happy to send this presentation. I know a number of people have asked if we could send the PowerPoint over. I'm happy to do that as well, if you guys are okay with it.
You're more than gracious. I believe we're recording it. And if you're gracious enough to answer those questions, I'll make sure they go out on our blog.
Oh, fantastic. Yes, I'll do it. I'm happy to do that.
Amazing.
Thank you.
The best topic there is, I think, and it's just so exciting. I always say you are not what you eat, you are what you absorb.
Yep.
And so that's the most fundamental thing in health. So, Karen, amazing presentation. You're a gentleman and a great presenter. You made it very succinct. Really appreciate it.
Thank you so much, everybody. Thank you so much for this opportunity. It's an honor.